So I studied the gut microbiome, this complex community of microbes in the distal gut. And I've done this for about the past 15 years. It's been an amazing thing to watch this field grow, our understanding excuse me, of this complex community and how it really connects to our biology. And my goal today is really to leave you with a better understanding of the basics of this field, where the field is at right now. But one of the amazing things about this field is that we're coming to realize that the reins are in your hands, that you can actually steer your inner microbes to a more healthy state by the decisions you make, by the foods you eat. And so I hope to convey that to you as well. Now, this is a huge field. I don't have time to cover all facets of it. And so another one of my goals today is to end in time so that we can have a nice discussion at the end. You guys can ask questions either related to my talk or to other topics about the microbiome, and hopefully I can answer those. Okay, so this cartoon refers to Easter Island, a landscape that was once covered in trees, and then through deforestation, introduction of invasive species, the landscape totally changed. And it's become kind of this prototypical example of what we now understand about ecosystems. If you change the species that inhabit that ecosystem, you fundamentally change how that ecosystem operates. So our bodies are ecosystems. We have microbes all over us. You think of us as just one species, Homo sapiens. We're actually an aggregation of thousands of species working in concert to create us, this superorganism. And so we have them all over our skins, in our nose. These microbes are crawling all over us. It's a little creepy, yeah. But most of them reside in your distal gastrointestinal tract, in your colon. And that's what I'm showing you here in this image. This is a cross-section of colon. So we actually take the colon, we section it, we stain it with different fluorescent probes, and then we look at it under a microscope. And that's what you're looking at here. So in the top right-hand corner, all these little fluorescent rods are different bacteria that live in the digestive tract. And then in the bottom right is, the top left, the bottom right is the host tissue. These are colonocytes, colon cells, the lining of the gut. And then there's this green strip running through the middle. Anybody want to take a guess at what that is? Mucus, that's right, that's mucus. So that's what the colon cells secrete to keep these microbes in the right spot. Most of these microbes have no intention of causing disease, and as long as they're kept in the right spot in the middle of the gut, they're happy, we're happy. You've heard the phrase, good fences make good neighbors. That's true also in the gut. But you can see immediately how dense this community is. You can see these microbes packed in there, the complexity. We've used different fluorescent probes to stain them different colors. You can see there's somewhat even of an organization to it. The little pink ones are clustered together. That changes if you change your diet, if you treat them with antibiotics. We'll talk more about that. But what we have here is an incredible interface for exacting change on our biology. These microbes are making chemicals, secreting them. We absorb them into our bloodstream, affecting our biology all over our bodies. So we harbor trillions of microbes. By cell number, we're more microbial than we are human. Think about that. You're microbial, carrying some human cells with you. The aggregate genome of all of these microbes is staggering, 100 times greater than our own human genome. So they bring metabolic capabilities, developmental signals that we haven't evolved on our own, that we've shopped out to these residents on us. And so the field is really working with two major questions: understanding who is there, what microbes inhabit different parts of our bodies in different biological states during disease, after antibiotic treatment, but also trying to understand what these microbes are doing. What are their dynamics like? How do they integrate with our biology? And so what are they doing? Well, they're doing a lot. We've known for quite some time that the gut microbes are involved in digestion, involved in gut health. This is fairly intuitive. This is where they live. They're actually doing much more than that. They're influencing our biology all over our bodies. They influence our metabolism. They can influence obesity. They can influence our immune system all over our bodies. How likely you are to fight off a respiratory infection, how fast an autoimmune disease progresses, how well an immunotherapy works can all be influenced by what lives in your microbiota and what they're doing. Our central nervous system is affected by these microbes too. We know that neurodegenerative disease, even moods and behavior in animal models can be influenced by changing the gut microbiota. So we're right at the beginning of understanding all these details, but there's tremendous potential here, tremendous opportunity, and a lot that we still don't understand. I wanted to share with you one pivotal experiment that occurred in around 2005, 2006 that changed everybody in the field's mind about how important the microbiota actually is in biology. And this was an experiment done actually in the lab where I was doing my postdoc. I didn't do this experiment, but I watched it happen. I couldn't believe it when I watched it. Researchers there were studying obese versus lean mice and studying their microbiota, and they saw that they were different. And it brings up this question, is the different obese microbiota contributing to obesity, or is it a result of obesity? This question of causation, very difficult to determine. And so the researchers there performed a really clever experiment. They transferred the microbiota into all lean mice. We call these wild-type mice, WT. They're just normal, healthy mice. So they're asking the question here, can the obese microbiota, if transferred into healthy mice, make those mice gain weight more rapidly? So these groups of mice were eating the exact same thing. And the white bar there, which is higher than the black bar, indicates that indeed the obese microbiota can make these mice fatter more rapidly. So that means the microbiota, when transferred, is conferring this physiological aspect on the host. Crazy, mind-blowing. And so since this time, these sorts of experiments have been done over and over again, showing this repeatedly. You can confer host phenotypes by transferring microbiota. Amazing. And so we have this microbiota that's wired into all facets of our biology. Here's the question. Can we create tools and a knowledge base to control these microbes in a way that optimizes our health, helps treat disease, helps prevent disease? And one of the great things about the microbiota, unlike our human genome, is that it's malleable. We can change it. So this is the future. The microbiota in precision medicine. It's malleable, we can change it. It's wired into virtually all aspects of our biology, and it's different in each one of us. And so that means that some day down the road, we don't know yet when this is going to happen, but it will happen, you will get typed when you go into the clinic. They will determine your microbiota type and then give you a treatment, perhaps even targeting your microbiota, that will help either treat a disease or prevent a disease. Microbiota-based precision medicine, and hopefully that'll be preventative medicine that will prevent diseases from happening. Okay, so the bad news, there's a problem. Our gut ecosystem is losing diversity. There have been many studies that have been performed on humans. We see there's a spectrum of diversity. If we were to map the microbiota of everybody in this room, there would be people with lower diversity and other people with higher diversity. There'd be a spectrum. In general, people with more diverse microbiotas, gut microbiotas, have better metabolic markers, better immune markers, less inflammation. But here's what's happened to our microbiota. In the industrialized world, we have all these great advances, wonderful food technology, great medicines, antibiotics, lifesavers, but there's collateral damage to all of these things, and that appears to be hurting our gut microbiota. What does this mean to our health? This is what we're trying to figure out. So at the same time that we're understanding that in the modern industrialized world, our microbiota is losing diversity, we also know that Western diseases are on the rise. On the left graph, this is from a very famous New England Journal of Medicine paper documenting the rise of autoimmune diseases. Things like multiple sclerosis, Crohn's disease, type 1 diabetes, asthma, all going up over the past half century. Same with obesity, projected to be 50% of adults in the US obese by 2030. Staggering. So could these be connected? So the way that medicine works in the Western world is very reactive, right? We wait for a disease to develop and we treat it. And this was born out of an era of infectious disease. You wait until a disease happens, you give that person antibiotics, and you fix that disease. This approach doesn't work so well with chronic diseases. Once they develop, they're very hard to get rid of. And so our approach has been, if you imagine Western diseases as this ugly tree growing over time, our approach has been to basically treat the symptoms, trim the branches. We see a new disease, we develop a new drug that can treat the symptoms, all the while the tree is growing. Another disease, another branch. Wouldn't it be more powerful to figure out how to stop that tree from growing? I like this cartoon. Great news, Captain. You can inform the passengers we have slowed the rate of sinking. There must be a better way, right? So chronic diseases are on the rise. What are the root causes of these trends? Can we figure those out? We have dozens of Western diseases all going up in synchrony. Are there really dozens of different causes of these different diseases? One of the amazing things about chronic diseases is most of them share inflammation as a common basis. That's the driver. And it may push me towards cancer, it may push someone else towards multiple sclerosis, someone else towards heart disease, but inflammation is the common denominator of a lot of these diseases. So could there be a few things that are causing inflammation in the Western world? Is there a unifying theory? And we actually think that diet by microbiota could be one of these. This is a hypothesis right now, but a lot of arrows are pointing in this direction. Okay, so let's talk about the microbiota in health and disease. We have to go back to just some basics here, basic facts about the microbiota. When you are in the womb, before you are born, you're actually sterile. You have no microbes in your gut. And then as soon as you're born, as you come through the birth canal, you're rapidly colonized by the first microbes that you experience. And then over the first months, years of life, as you interact with people, the environment, you acquire more and more microbes. These are the founders of your internal ecosystem. This is what sets the stage for a lot of your health. Immune trajectories, metabolic trajectories, influenced by these microbes that first colonize your gut. And this isn't just a random collection of microbes that we acquire. We've been transmitting microbes across generations for millions of years, hundreds of thousands of generations. So these microbes have co-evolved with us. They've undoubtedly shaped our genomes. So what does it mean for our biology when we've done this for millions of years, harbored these microbes, and then we start to lose species? It's as if we're losing pieces of our biology. This is a great image by an artist named Banksy. It really captures, I think, the main idea here. Could there be an incompatibility between our human genome and our microbiome? We know we have these two parts of our biology that have to work in step, our human genome, our gut microbes. But they have asymmetric plasticity over evolutionary time. So the microbes, that means the microbes can change really rapidly. Day to day, they can change. Week to week, certainly generation to generation, where our human genome stays relatively constant. And so what does it mean if you start to introduce big changes to your lifestyle that change this community? Could you end up with a microbiome that's fundamentally unexpected by your human genome, something that doesn't work as well? Now, one of the major changes that's happened over our recent evolution, over our recent time as humans, is a great decrease in dietary fiber. This is one of those boring aspects of diet that nobody likes to talk about. This is the fuel for the gut microbiota. And over the past 10,000 years, as we've moved from foragers to couch potatoes, we have decreased our dietary fiber from 100, 150 grams of dietary fiber per day to 15, average American intake. Incredible change. We know if we do this experiment in animal models, the microbiota is just totally different, totally changes. You lose species, they go extinct. So, why is dietary fiber so important for the microbiota? Well, it's where they live. So this is just a schematic of the small and large intestine, a cross-section. The microbes live at the distal part of the gut in the colon. And so food enters from the left over there, from the stomach into the small intestine. And if you eat something like cake that's filled with sugar, sucrose, starch, we have digestive enzymes to degrade these carbohydrates. We don't have many digestive enzymes for carbohydrates, but we have the ones that can turn these carbohydrates into simple sugars. We absorb those into our bloodstream, leaving our microbiota starving. If we eat something like broccoli, there are complex carbohydrates there. We don't know how to degrade these with the genes that we have in our human genome. They pass through the small intestine, arrive at the gut microbiota that is well equipped to degrade all these complex carbohydrates. 60,000 to 100,000 genes for degrading carbohydrates there. That's the profession of this community. And they ferment these carbohydrates into chemicals that then we absorb into our bloodstream that regulate our immune system and metabolism. So this is why dietary fiber is so important for fueling the gut microbiota. And so we're curious about what our microbiota used to look like when we ate so much fiber. And so wouldn't it be great to have a time machine where we could go back in time, sample different humans, different parts of the world, and understand what the microbiome used to look like? So we can't get that grant funded that will build us a time machine. But we can go and study traditional populations of humans that represent what the human microbiome may have looked like before we've modernized. So these are the Hadza hunter-gatherers of Africa, a really special hunter-gatherer population that's been very gracious in agreeing to participate in our research. And they represent this period of human evolution that we existed in for 95% of the time that we existed as humans. That period of time labeled hunter-gatherer up there on the timeline. From about 200,000 years ago to about 190,000 years ago, or till about 10,000 years ago, 190,000 years, we were hunter-gatherers, living like these people. They're a continuation of humanity's ancient past. And they eat huge amounts of dietary fiber. This is them cooking a tuber over a fire. The tuber there looks like one of the logs that they're fueling the fire with. And there's a image there of the tuber actually after it's been cooked, incredibly fibrous, very different than the tubers that we eat nowadays, right? And so we've been studying their microbiota. We see incredible patterns of seasonality. They actually have microbes in their guts that disappear in one part of the year and then reappear the next year as foods are reintroduced because they eat seasonally. They have incredible diversity in their gut. And then we see species there, microbial species that we just don't see in anyone in the United States or other industrialized countries. So, a lot of these studies have been done of different traditional populations. The data's scattered about. I had a wonderful student who graduated recently, and he decided to bring all this data together and put it onto one chart, see if any patterns emerged. And that's what you see here. So on the left are labeled all the different populations that he brought in. This represents their microbiome composition. And then he arrayed it on this x-axis. So the bar there represents the distribution of composition of their microbiota. Basically, where that bar is represents their microbiome and what populations it's like. And so you can immediately see a pattern. There's this group of bars on the left and a group of bars on the right. And the pattern there is traditional populations and industrialized populations. Low diversity, high diversity. Okay? So we're all low over here, they're all high. And so it doesn't matter if you live in Africa, South America, Papua New Guinea. If you live a traditional lifestyle and you haven't been embracing this industrialized way of life, you have the same species in your gut. These are populations that are separated by tens of thousands of years of human migrations. They have the microbes that have been marching in step with us, and we don't have a lot of these microbes. So this is the metabolic switch that we've undergone. All these populations have a lot of diversity in their microbiota. They produce a lot of chemicals like the short-chain fatty acids that go into our bloodstream. We have a lot of blood sugar. Very different metabolic shift there. What does this mean for our biology? In addition, as our microbes starve and don't have dietary fiber, guess what they start eating? Yeah, they start eating you. Your mucus. That mucus layer, that green layer, is made of carbohydrates. And it's a backup food source for these microbes that don't have anything else to eat. So we were curious, what would happen in a mouse model if we fed them high and low fiber? What would happen to their mucus layer? This is not an easy experiment because you have to quantify microscopic images. Luckily, we have a tremendous bioengineering group here, and collaborating with them, they automated the image analysis. And I'll just show you some representative images. This is the mucus layer, the green layer there on the high fiber diet, pushing away the red microbes from the blue host tissue. The yellow there is actually plant cell wall material. This is part of the high fiber diet. This is what happens on a fiber deficient diet. That mucus layer thins, the microbes move closer to the epithelium, and markers of inflammation start to be induced. This is just after a month in a mouse model. Imagine decades or generations of this. Okay, so that's all very bleak. Is there hope? There is hope. There is hope. So what can we do now? So the amazing thing, I work with my wife. We're both microbiome researchers. We run the lab here together. And she and I, on this journey of learning about the microbiota, started realizing that we were living our lives differently, eating differently, raising our 2 daughters differently. And none of our friends were doing this, whether they were scientists or non-scientists. They were kind of not on the same journey. And then we go to a microbiome conference, and everybody was doing the same things we were doing. And we thought, well, we just have access to information, this privileged access that other people don't know about. And so we thought, well, we need to write a book that will help convey this important information, make it available to everybody. And so that's why we wrote The Good Gut. It's really to tell about the science in this exciting field very broadly, but also to tell you about the journey that we've been on for changing what you do in your life, what you can do to change your gut microbiota, make it healthier. So we got written up in New York Magazine, and the journalists that covered they actually came and did this photo shoot in our house. They spread manure on our dinner table, which we were fine with. This journalist wrote at the top there, the quote, "They're making a commitment to fiber consumption today that borders on the comical." And what I really think is comical is still how far even my family is from eating these ancestral levels of dietary fiber. We come nowhere close to 100 to 150 grams per day. We get about 35 to 50. That's doing well. It's really hard to eat that amount of fiber. But there's really no controversy about this advice. If you go to the major authorities, this is the Dietary Guidelines Advisory Committee. On the top there, intake levels of dietary fiber are half the recommended levels. We need to eat more high-fiber cereals, whole grains, fruits and vegetables. Harvard School of Public Health, fiber is an important part of a healthy diet. You should get at least 20 grams a day. More is better. Nobody's documented an upper limit for fiber for health. Mayo Clinic, very simple. Eat more fiber. Now, they don't tell us why, because this is all based on epidemiological studies. We know if we look at large populations of humans, the people that eat more fiber are healthier, and they're less likely to die from all causes. And there's a dose response. Increase 10 grams per day, you decrease your chance of dying. Amazing. And while they don't know the mechanism for why fiber might do that, I think the microbiota is giving us some insight into what the potential mechanism might be. So in addition to the book, we've also written smaller articles. If you're curious in just reading something that's bite-sized, this is a piece that my wife wrote, Erica, 5 ways to boost your kids' gut health. Feed your microbes. We've talked about that. Eat bacteria. There's a lot of evidence that the bacteria in fermented foods are very healthy. Don't over-sanitize. So during cold and flu season, yes, important to wash your hands. After petting your dog, maybe not. After playing in your lawn, playing out in your yard, if you don't spray chemicals around, maybe not. Avoid unnecessary antibiotics. Again, wonderful drugs, but they're overused. We need to have that conversation with our doctor. Are antibiotics really necessary here? Can I take a wait-and-see approach? Expose yourself to nature's microbes. Get out into nature. Experience environmental microbes that are healthy, not the ones that you find in the subway, but the ones you find out in nature. So this is a case study, me, what's happened to my microbiota while I've increased my gardening, increased fruits and veggies, let my kids play in piles of manure, eat pizza that's made of whole grain crust, sourdough, fava beans, onions, kale. That's a delicious pizza that will also feed your gut microbes. I culture my own kefir. You don't have to do that. You can buy it. But it's easy to do this. If you can get kefir grains, you can order them online. Just strain it, new milk every night. You have a delicious beverage in the morning full of healthy microbes. And then get a mangy dog and play with it as much as possible. So what's happened to my microbiota over this time? These are the number of species, 2010, 2015. Gone from about 600 up to around 1,000, 1,100. I have no idea if that is related to my lifestyle changes. N equals 1, who knows? But other studies have documented that if you change diet, you can increase microbiota diversity. And here at Stanford, we've started the Center for Human Microbiome Studies that I co-direct with David Relman. And our goal is to really start doing experiments in humans where we change diet in a positive way, see how that changes the microbiome and how that changes health. We have a wonderful network of investigators that are supporting this. Kari Nadeau, some of you may know, she studies allergies here. She's wonderful. We're enrolling some of her kids in our studies. Mark Davis, world-renowned immunologist, has brought human immunology to Stanford. And so now not only can we see if we can change the microbiome of these people, we can see if we can decrease that simmering inflammation. And then Christopher Gardner, who's speaking next here, is really the key to this whole thing. So he knows how to get people to eat differently, free-living humans. And it's not an easy task. We're wired to eat sugar, fat, salt. That's what our brains tell us. If you go to the Hadza, they don't like eating their fiber-rich foods. If you ask them what they'd prefer to eat, meat and honey. Two components of their diet that they can't get very often, but when they get it, they eat as much as they can. If you put them in the grocery store, in a restaurant, they would make the same bad decisions that we make because that's the way our brain is wired. But Christopher has figured out how to get people to eat high fiber. And so he's the key for running these dietary interventions. Finally, I just want to thank my research group. A lot of the ideas and data that I showed you today is from this wonderful group of graduate students, postdocs, other scientists. You can see our dog there, even part of the group. My wife and I have a Facebook page where we cover the microbiota, send out information about new discoveries and what they mean. Feel free to sign up for that. And then also thanks to funding sources. We've had wonderful financial support from the NIH, from Stanford, and from Burroughs Wellcome Fund, and that's really a key to doing this research. And thanks to all of you for your attention. I'm happy to take questions. Yes, please.
Thank you so much. I think all of you are on our own.
Yeah.
What would you do if you were us?
Yeah, it's a difficult situation. So the question is the disconnect between what I'm telling you here and what the medical community knows and conveys to patients, I think, generally. And when you leave this room, what are the steps?
Even if we have Stanford doctors.
No, I know. Yeah, I agree. So I would say I know a lot of the Stanford doctors. Many of them have read our book. I've had conversations with them. They're becoming very enlightened. I'd say among the most enlightened in the nation on this topic, this important topic. But it's difficult because we have dietary advice now. But what's been documented is that doctors don't have success in getting people to eat differently, and so they've given up on it.
What about just testing? Are we all on our own to go out and do testing?
Yeah, so there are not easily accessed tests for the clinic. There are services where you can send out samples to companies. You can look into those and survey your own microbiome. That's the best option right now. We're hoping this human center that we've started will move at a pace where we can rapidly get something going that will allow us to profile patients where we think the microbiome may be a key element of their disease, but we're still a little ways from that. It's a good question. Yeah, please.
Talk about prebiotics and probiotic supplements and if those really work to help increase the diversity.
Sure. The question is about prebiotic and probiotic supplements and whether those help to increase diversity. I think there's a lot of potential with these supplements for helping people. The lack of regulation in the supplement space puts a lot of products on the shelves that are of poor quality. And so we actually have an entire chapter. I'm not trying to sell more books, but we have an entire chapter that we call where we call these probiotics transients, and how to navigate that space and find good options. So there are independent organizations out there that will test probiotics, and you can see the certification on the outside of the probiotic label. And so I would look for those. Look for these independent entities that are certifying the probiotic products. Prebiotics, it's a similar state. Prebiotics are basically just dietary fiber that's been purified. And what appears to be the case is that a mixture of dietary fiber is much better than a single prebiotic. And so the best way to get a mixture of prebiotics is to eat diverse plant foods. If you can't do that, you can supplement your diet with diverse prebiotics, but the science is still very unclear how these compare with eating real foods. This is one of the studies that we're conducting actually to try to figure this out. But I like fermented foods and eating plants as the options for getting live microbes and fiber into the microbiota.
Two questions. Great talk, great book, by the way.
Thanks.
How is the connection between the strength of the immune system and the health of the microbiome? How was that discovered?
So the question is the connection between the microbiome and the immune system and how that was discovered. So there have been many studies in animal models to start with that show diverse effects of changing what's going on in the gut microbiota, changing other things that are going on in the animal's body. So for instance, investigators that have a lung infection model have shown that if you go in and change the gut microbiota, that lung infection will behave totally differently, either progress more rapidly or not. This has also been shown for autoimmune diseases. They'll induce a multiple sclerosis-like syndrome in mice and show that by changing gut microbes, you can change the rate at which these diseases progress. And this has been shown for immunotherapies. So a lot of this is mouse models, and the question is, how does this apply to humans? And we know generally the principles are likely to. What is true for the mechanisms? What we know is we can go in and mess with the microbiota by diet, change microbiota diversity, and you see immune markers change coincident with that. Yeah.
Thanks for your talk. If someone that you are constantly is constantly in your life, physically close to you, and they are sick, do their microbes jump to you, and do you get sick?
Yeah, very, very interesting question. So so did everybody hear that? The question is, if you are around somebody that's sick, can their microbiome invade your microbiome and make you sick? And in fact, it's even been pointed out, knowing that the microbiota associated with obesity is different, that if you look at obesity rates around the country over the past decades and their increase, it is actually spreading almost like an infectious disease, the pattern. And it leads to this question of, could it be obese microbes that gradually are spreading around the country? There's experimental evidence that you can override this if you eat a healthy diet. And in fact, a diverse microbiota can invade a non-diverse disease-associated microbiota if the diet changes to support that diverse microbiota. So we think that diet is really the key here in determining what microbes do or do not invade. So the question is, where is the inflammation that happens? The immune system is everywhere, and it can be at a specific site. Mechanisms will vary, but in many cases, it is everywhere your blood goes. Okay, so these cytokines, these chemicals, the immune cells, they circulate through your whole body.
Yes, you had mentioned something about inflammation being a root cause. That's through there. So for evidence-based, we've been looking at the CRP, C-reactive protein.
Yeah.
And I've been checked for that, and I mentioned it to my doctor at Kaiser, and he said, yes, you can tell, but there's no way to know where the inflammation is. I wonder if you have any expertise in that area.
Well,
Thank you.
know, yeah, no, great question. So a C-reactive protein is one of these proteins that's been used as a marker of inflammation. It's associated with heart disease and potential risk of cardiovascular health problems. An amazing study just came out in the Chimani, a traditional group in South America that has incredible heart health. They've imaged their arteries, just very few atherosclerotic plaques that we try to avoid. They have really high levels of CRP, of C-reactive protein. And so it may have to do that with lifestyle and diet overriding how important that is in determining cardiovascular health. But in the context of the Western world, it's definitely well recognized that we try to keep CRP as low as possible. And so how to do that is probably a mixture of the things that I talked about.
So
Sorry.
do
Yeah.
GMOs have any effect on your microbiota?
Yeah, do GMOs have an effect on the microbiota? That hasn't been looked at in great detail. I get that question a lot. I think it's a good question. GMOs is a very broad spectrum of products, and really we need to do a few good studies to look at that. We don't really have any good information.
Hello, yes. Thank you for your talk. And those of us in the autoimmune community that haven't had luck with conventional medicine have looked a lot at the leaky gut syndrome. And whenever I talk to doctors like the other lady, I've had disappointing feedback, but through the blogs. And can you tell us about leaky gut syndrome?
Yeah, sure. So the question is about leaky gut and its association with autoimmune diseases, inflammation. This is something that has been talked about widely, and the data is coming along, but I would say it's one of the parts of microbiome science that's really lagged, and I think a lot of people didn't believe it for many years. It's the idea that these cells that make up the lining of your gut, basically the grout that seals those tiles together can break down and let things across the barrier into your bloodstream that shouldn't go. And can induce inflammation. And there's great evidence that in animal models you can totally change how permeable the gut barrier is by changing the gut microbiota, through diet. So there's good evidence that this is modulatable through the microbiome. And I would say that the data that I showed you on the thinning of the mucus is good evidence that there's a breaking down of the barrier due to low fiber diet. And so I think if I were to recommend to somebody if they think they have leaky gut or have some clinical evidence of this, try gradually ramping up your dietary fiber to see if you can refortify that barrier.
Can you tell us what you've learned about a more recent research on the effect of artificial sweeteners and gums on the microbiota?
Yeah, sure. So gums specifically haven't been looked at in great detail, but artificial sweeteners have in a couple studies. And there's indication that, and this was a beautiful study published in Cell a couple years ago, that different artificial sweeteners in 3 different classes can negatively impact the microbiota and lead to changes in metabolism that are similar to metabolic syndrome. So there's evidence that these artificial sweeteners can negatively impact the microbiota, and that can go on and negatively impact your health.
Can the way you cook your food, whether you microwave or actually steam it, does that affect the the microbiota? Because I've been told not to microwave my food.
Yeah, interesting. So there, I've talked a lot about dietary fiber. There are a lot of other really helpful compounds in a lot of the plants we eat, and those may be susceptible to breakdown due to superheating or different ways of cooking. I would say that, though, is generally as Americans, if you can figure out a way to eat more plant-based food, whether you microwave it or steam it or stir fry it, that's not going to matter. What's going to matter is increasing the amount of plants that you take into your diet. Yeah.
Hi, I have rheumatoid arthritis and have spent 4 years digging into all this stuff. But one piece of conflicting information I get is actually whether I should do probiotics or not. I'm also allergic to yeast, which doesn't help. But I do it. But the bottles and all the stuff always says, if you have an autoimmune condition, you may not want to do probiotics.
Yeah.
Can you talk a little bit about that, of whether I should or not, and how do I approach figuring that out? Because of course the doctors don't know anything about them.
I mean, in terms of specific probiotics and how they impact specific people with different microbiotas that have specific indications for why they would be used, we are in the dark ages. And the way forward, I think, is to look if there's been a really nice controlled study that looks at people with your condition, and if there's a probiotic that appears to work, try that. But even then, because we're all so different, I think that it's a trial and error situation. Probiotics generally are used with the idea that they probably won't hurt and they may help. And so you just have to survey, yeah.
Hi, thank you for your talk, and want to hear from you of how the microbiomes change with antioxidants, food like berries, and also want to hear from you about organic foods,
Yeah.
Different ethnicity diets like Mediterranean diets, Asian diet, they
Yeah.
are more plant-based.
Yeah, these are great questions. So she's asking about different facets of diet, antioxidants in food, and different cuisines and how they may differentially impact the microbiota. And I think that these are the types of questions that we hope to get to with the Human Studies Center. We just don't have a lot of information on this yet. We know that besides fiber, there's a lot of compounds in plants that when we consume them, the microbes actually metabolize, change their chemical nature, and then get absorbed into our bloodstream. We have no idea what these compounds are doing, and so this is something that we just need to study in more depth. Oh, yeah.
Yes, so I have patients that have SIBO, and so sometimes there's this discussion that if you give them probiotics, it might colonize in their small intestine.
Right.
So I'm a little hesitant to prescribe that, but at the same time, I'm curious about the enema probiotics, if that could work for them, and then also fecal transfer, why that's only for C. difficile. I know
Yeah, great. Okay, so 3 really important questions. So the first one is about small intestinal bacterial overgrowth syndrome, known as SIBO. And this is the idea that microbes are kind of migrating up your gut into the small intestine. There are too many microbes there, leading to big problems associated with metabolism of a lot of these simple sugars and fats and things like that. I don't know of any evidence of probiotics colonizing in the small intestine to change effects of SIBO. There may be a study out there. I think that this is one of these things that is probably solvable through diet in the microbiome, and it's something that we just have to design some good studies for. There's not a great set of data for addressing what you just asked about SIBO. But again, this is a growing problem. More and more people are getting SIBO, and it's something that we're going to have to start studying in depth. Fecal transplants. So people have probably heard of fecal transplants. If you haven't, they're exactly what they sound like. And they've been a miraculous cure for C. difficile colitis. Over 90% cure rate. If you have C. difficile colitis that's recurrent, getting healthy stool that you administer through a clinic, as opposed to 25% cure rate with antibiotics, which was the standard of care before this. So a miraculous cure rate for fecal transplants for C. diff colitis. There are over 120 open trials right now at NIH for testing whether fecal transplant can work for a variety of other conditions, ranging from arthritis to inflammatory bowel disease. I mean, they're testing everything. I haven't heard of anything where the success rate is like C. diff colitis, and my guess is that because we move in a stepwise fashion in science, oftentimes dietary support of the incoming microbes is not provided as part of these trials. And I think you have to support these healthy microbes that come in with the right diet, and that will be the next big piece of the puzzle, I think. Yeah.
Do common diabetic drugs like metformin and glipizide affect the microbiome?
Yep, absolutely. So there have been some really nice studies looking at this. Metformin is a drug that is not supposed to have a direct activity necessarily on the gut microbiome, but it has big effects. There are many groups that are looking into this right now to try to better understand that. Yeah. Unclear.
So I have a question about the fiber data, which I found kind of fascinating. So 150 grams, at least to me, because I don't know, doesn't sound like a ton. And I used to think I eat a lot of fiber. What's eating 150 grams of fiber look like? Before you eat it, I should qualify.
Yeah. It's great. I tried to convince a journalist at Time Magazine. So the question was, what does eating 150 grams of dietary fiber look like? I tried to convince a journalist at Time Magazine to do one of these personal journeys where she was trying to get... Yeah, like Supersize Me, but in the other direction. What does a shopping cart look like for a week if you're trying to do this? What is making dinner? How much time do you have to dedicate to chopping? And then what does your plate look like at each meal? I don't have a good... So it's incredibly hard to do, I think, because we have bred plants to be rich in sugars and simple starches, and bred out of, because these tubers haven't been cultivated, what we're left with is a potato that doesn't have a lot, that only has really fiber in the skin. And so I think it's probably something that's somewhat unattainable unless you're solely focused on that. These hunter-gatherers, in fact, all they do basically is hunt and gather all day, and they're eating. So they're just fluxing massive amounts of plant material through their bodies. So you'd have to do something like this, but probably even more so because of the low amount of fiber in the stuff that we've domesticated. So I don't have a good, that size. Yeah.
So does it matter if you blend the fruits and vegetables into a smoothie, and then also raw versus cooked?
Yeah.
Fresh, how fresh do things need to be?
Yeah, great question. So we think that raw versus fresh is probably really important. I mean, all human cultures, unless you're a raw foodist, they all cook. And so it's, I think, generally accepted that cooking is just something that is likely to be compatible with good health because we've done it for over 100,000 years. But the amount that you cook something and how you cook something is likely to drastically change how accessible the fiber is to the microbes in your gut, something the field hasn't even come close to addressing yet. Would be very interesting to look at. And then this idea of blending in smoothies or how well you chew something undoubtedly also totally affects this. If you think about flour, a grain of wheat is endosperm, all starch, wrapped in this bran layer. And so when you think about it, that's really a nice delivery system for getting fiber to your gut microbiota, surrounded in this bran layer. If you grind it up into infinite fineness for bread, you make all of that endosperm available to be digested in your small intestine and go in as blood sugar. And so there's probably something really fundamentally connected between how well something is ground and how little of it reaches your gut microbiota. There'll be exceptions to that. You can take complex carbohydrates that we don't have the ability to digest, and no matter how much you grind those up, they still won't go into our bloodstream until the microbes get to them. But other things like this wheat example, it totally matters. Yeah, so it's a similar idea. There are probably inaccessible sugars due to the structure of the material, and the finer you grind that up, the more you make that accessible.
Years ago, I read a suggestion that there are lots of kinds of bacteria that are not cultivatable on plates, and so they were unidentified. Do you have a way of identifying?
Yeah, great question. So this is kind of a sciencey question. So he's asking about studying these microbes in the laboratory. We normally grow them on petri dishes or plates, and And so a lot of the microbes in the gut, we don't have a good way to cultivate in the lab. The way that we study them is through DNA sequencing technologies. We can take any material from anywhere, the ocean, a rotting carcass, or our digestive tract, and grind it up and purify out the DNA and sequence that, and then identify what microbes were there. We still don't have the ability to cultivate after that. That doesn't give us the ability to study them in detail in the lab, but at least we can define who is there based on that technology.
Is there any connection between like cancer, like colon cancer, and what you're talking about? And also, do they teach dietary in medical school these days?
Yeah. So the medical school education is an important one, and that's something where, again, Stanford is on the cutting edge, revamping the curriculum here to really make sure the med students are getting good information about both the microbiota and about diet. Related to, is there a connection between the microbiota and colon cancer? Absolutely. There's a beautiful study done where they actually took African Americans and basically put them on an African diet, and they took Africans and put them on an American diet. And over a very short time period, I think like 6 weeks, they saw the markers that are indicators of the likelihood of development of colon cancer totally change in these groups, just based on diet. Because the observation was made that African Americans have a much higher rate of colon cancer than Africans living in Africa.
Dr. Sonnenburg? I've been reading a little bit about paleo diets, and they talk about inflammatory foods versus not.
Right.
And they say that dairy and any kind of grains are considered inflammatory, especially wheat. Can you comment a little bit about what that has to do with this?
Yeah, so that's a really complex, and this gets to non-celiac gluten sensitivity. How does a paleo diet interact with all the information that I told you about the microbiota? So I think the concept of the paleo diet is a great one. I think how it's implemented is a big problem. I think people associate it with a lot of meat and avoiding a lot of plant-based foods, because things like legumes and whole grains, because they came about after the dawn of agriculture. That's actually untrue. We were eating grains well before that. It's just they weren't formally cultivated. We were collecting them and eating them. But I think that the paleo diet in spirit is the right way to think about it. What does our body expect to be eating? Now, in the case of non-celiac gluten sensitivity, this is something that I think is a big mystery, why this is going up, why can't people eat gluten, and could be related to increased inflammatory state, that suddenly we become more reactive to things that we shouldn't be reactive to. And so I think it more likely reflects a misregulated immune system than something that's happened in our food chain. I also think that usually when people eat wheat, they're eating crappy wheat that the bran has been stripped from and has been ground up. And we know that this basically just increases blood sugar levels. And so I think that there's a connection between feeling bad and wheat, when it may not necessarily have to do with wheat, but it just has to do with eating refined products.
I have a two-part question for you. I was wondering if certain diseases have been linked with the diversity of certain microbiota, and also, is the biology of each microbiota the same with respect to, are certain fibers more favorable to certain microbiota over others?
I say that last part again, are certain...
Like fibers more favorable to others?
Yeah, great questions. So certainly there have been studies associating better diversity with better health. How we get to that with these different fibers is something that we're exploring right now and trying to figure out. We have grants pending right now to look at, for instance, one prebiotic fiber versus a mixture of 7, to ask this question of, is diversity of fiber something that's important in cultivating diversity in the microbiota, or can you do this very simply by just eating Metamucil or inulin? I think that's probably unlikely, but we're going to test it formally. And in terms of whether there are better fibers or worse fibers, there probably are, and it probably gets back to structure and what foods they occur in. We have very little information about this right now. The information that we do have is that we can just broadly benefit by eating more diverse types of plants. And that's where we're at right now.
I have a question. Supposing you have to take antibiotics, I have to take them for a couple weeks. How do you replenish the destroyed, devastated biota system? Does yogurt help? I mean, what can you do?
Great question. So after antibiotics, again, we want to look at this more formally, but evidence suggests that yogurts and probiotics definitely can help reduce the, for instance, occurrence of antibiotic-associated diarrhea. And so they probably don't permanently colonize, but they can serve as placeholders while your resident microbes regrow. And then just based on what we know about the microbiota, I think there's an assumption, but again, to be formally tested still, that eating dietary fiber that would fuel the regrowth of these microbes that have been hit so hard by the chemicals is probably likely to be beneficial in helping the community recover.
Thank you.
Yeah, sure. All right, thanks everyone. Yeah.